Thank you, Rich.
David and turtles.
All right, so I tend to talk fast, so I'll try to slow down like a turtle for this one. Okay. Hi, everyone. My name's David. I'm an undergraduate student from the University of Southern California, and today it is my pleasure to be your guide to talk about my project and the journey that it was to create the first drafts of the genome for Trachemys scripta elegans, or the red-eared slider, with the goal of basically having this genomic foundation so we can understand hybridization and introgression that we actually just, a current study, discovered that these cute little guys have been undergoing. And well, if they look familiar, it's for good reason. Basically, they're super common, both as pets and now in the wild. In fact, if you go outside in the California Golden Gate Park, the ponds and slow-moving water habitats, I pretty much guarantee those are the only turtles you're going to find at this point, mainly because they were once pets. They were involved in the pet trade of about, between 1989 and 1997, about 52 million specimens were taken where they were farmed and then sent out all over the world. You can buy them in Chinatown, these cute little guys. They start this big, live for about 70 years and get about yay big. So whether you get bored of taking care of them or you go to college or move, people think it's a really nice thing to do to let them back into the wild, but that's not where they belong. And as a result, over that time, they become an invasive species and outcompeted a lot of native populations, both turtle and non-turtle, in fact. So yes, you'll find them in ponds. They're easily identifiable. They're called the red-eared slider because they have that little red mark, as you can see. And native to, again, slow-moving freshwater habitats between Alabama and northeastern Mexico. But now, because of the pet trade, they have been found basically everywhere, all over North America, Europe, and Asia. In fact, they are such a problem. I do know that some European countries have banned the import of these turtles because they're just doing so well everywhere they go. So how are they doing so well? Well, this study helps to find and address that question, but I guess it's kind of nice to go back to the start. Basically, a couple years ago, there was an expedition to Pecos River by Brian Simonson, my advisor for this project, and other of his colleagues. Pecos River. So we all know about the Rio Grande, a large river running through Texas and bordering Mexico. And then a little bit to the east of it, we have a tributary, the Pecos River. And on the Rio Grande, we have a different population of turtle, Trachemys gagei, located in this little blue spot over here. And these guys are quite different, actually, from the Trachemys scripta elegans, the red-eared slider. These are the big bend sliders, and probably the first thing you'll notice about them is they're really buff. Because these guys, they prefer to live in fast-moving, flowing river environments, so they have their large arms. We think, basically, well, it's easier to navigate it and fight against the force of the current. And compare that to the native range of T.S. elegans, the red-eared slider, again, from Alabama to like northeastern Mexico, and comparing what we saw with the Big Bend with the red-eared slider, these guys all look scrawny. I mean, look at those arms; they can't do anything with that. But what's interesting, this one came out as a surprise. Individuals that were sampled along the Pecos River, again, only Trachemys, as far as we know, live here. There are no T.S. gagei, Trachemys gagei that live here. Individuals that were taken here and then genotyped, as shown as such, individuals shown in that box right there, they were exhibiting actually markers from Trachemys gagei, which don't even live in the Pecos River, which is really bizarre. We're not really sure how it happened, but basically they found evidence, rather, I didn't do this, they found evidence of hybridization and introgression, and which is a very interesting discovery because it leads to the question, or it leads to the hypotheses, rather, that we know T.S. elegans is invasive, and here we have a possible explanation for the mechanism, the molecular and genomic mechanism, how they become, and how they are so successful and invasive. So, several hypotheses. Well, humans like to dam up rivers, and whether it's for hydropower or for reservoirs and whatnot, and that changes your fast-moving river environment that, for example, Trachemys gagei might want to live in, and instead creates them a much more slow-moving that would be more favorable for the red-eared slider to live in. And of course, this introgression. What we would like to do, or when this is where I come up, what we would like to do is get a more big picture understanding of the genomic and molecular mechanisms for introgression. So to do that, we went through a whole genome sequencing approach. And that was done before I came here, thank goodness. I don't know how I would have managed otherwise. The time it takes to get a genome predicted to be about 2 billion bases long. So anyway, how the genomes were, how you sequence a genome. So first, they went out, they collected them again from the Pecos River. They collected tissue samples, actually came from one individual. Again, this is a trachomys, identified morphologically with its red ear. And then basically the DNA was extracted, and the DNA libraries were also prepared here in the California Academy at the CCG. And then the actual sequencing mostly was sent out to Illumina, where they did all the next generation high throughput sequencing, etc. So cool, that's all nice and done. And I want to talk about a very brief understanding of how the data looks like, because that will make sense later on, and how to process it. So a lot of you have done Sanger sequencing, and we've seen some examples of them today, where you basically identify certain genes, you sequence from one end, sequence from the other end. It's about at least 1,000 bases. I don't know how long your guys are exactly, but basically look for consensus and overlap, and that basically reports your sequence. Well, what you do instead, when you want to go for a whole genome, that would take way too long and be way too expensive. So to represent DNA as bars instead of helices, basically you blow it to pieces using sonication. And then these are your libraries, you modify them, tag them, so you know which one's which, basically. And what's important to notice is that all these reads, which look like a mess, that's exactly what they look like. A lot of them will be copies of each other, because again, when you extract your DNA in your genome, you have multiple copies of it. And then basically you send these reads to Illumina and they sequence them, these fragments, basically. So you have a physical piece of DNA called a fragment, you sequence them using Illumina next-gen sequencing to get a computational sequence, which we call a read, about 150 to 300 base pairs, depending what kind of method we choose. So those are how our libraries look like. It's basically a big mess. So then I come in and I'm like, okay, how are we gonna assemble these? Thankfully, the folks out there have done it before. And just to get a quick specs, our sequencing runs had a total of about 150 million of these 150 base pair reads. So yeah, how are you gonna put it all together when it's a big mess like that? Well, luckily, there have been published programs that have done this. These are the two I used. The key takeaway here is that they both have short sequences and short oligonucleotide. Basically, they're designed to deal with these really small 150 base pair and 300 base pair sequences and string them together to make assemblies of whole genomes. So this is going to be fun. We're going to talk about actually how this algorithm works. So as an example, let's say we have a read. This is 13 bases. And the most important parameter when you are assembling a genome is selecting a k-mer size. Quick pause. A k-mer is basically all possible subreads, subsequences of length k that can be reduced from a read, where k is an integer. So for this example, we're demonstrating this in this 13 base read. We're gonna, or is it 10 base? I don't know. We're gonna choose k size of 3. So computationally, you have all the reads, and the computer basically splits it up like this. Okay, so all these subsequences, they all align with each other to stack up to make the full sequence. Okay, so first you split into k-mers. Here we have a 3-mer, and then these programs, they basically take all your k-mers from all your reads, all 150 million of them, and sort them into nodes, okay, which I choose to represent with this little graph right here, so we can see it. And then basically now we got to walk through the node, and the trick is that you want to start at the start. Here we have a TAC, and you want to end at the end, at the ACT. And again, the trick is that you want to walk a path from start to finish, touching every single node. If you leave one out, that means you missed something, basically. And how do you touch a node? Well, you make an edge, where an edge is basically defined as when you have an overlap between two nodes that overlap by k minus 1. So in this case, we have k-mers of 3, so we want to overlap of 2. So this TAC has a nice overlap with this AC, so you make an edge, okay? And then you add on the next base G, okay? Let's find the next one. It goes this way. And it's an iterative process, basically, until you touch every node, and that builds your sequence down there. And you get from start to finish. However, this is a little reductive. We actually didn't use 3-mers. As you could probably guess, that would produce tons of ambiguities and uncertainties about what is the next proper path. So in this demonstration, we had 3 mers, we only used 1 read, and they're about 13, this one, was 13 bases long. For an actual assembly, we were using k-mers all the way up to 128, like sub-reads that we had to get that overlap of, so an overlap of 127 bases. We had, again, 150 million of them. Basically, it was a lot bigger. And I want to emphasize again, this k-mer size selection is, I would, from my understanding and my experience in this, the most important step in doing your assembly, because if you get it too small, it's easy to get an overlap to have a nice contiguous assembly, but as we can see right here, you might have an ambiguity. You don't know, does this go this way or does this go that way? So to resolve that, you can increase the size. Sure, you'll get fewer paths and you might miss some, but it'll resolve the ambiguities, but it'd be hard to find overlaps. You might have gaps in your sequences. So this is all nice. It only gets you so far though, because real life is it's impossible. It's impossible to guarantee that you can get from one end of the graph to the other and touch every single node. That's just computational when you're talking about these huge numbers. So not only that, the actual sequencing method that we talked about, blowing it all up, it's inherently random, so sometimes you might not have enough coverage, you might just not have the data, you just might have gaps. Or sometimes you might make a mistake. So don't need to know what they say, but here we have basically 2 graphs, and I spent time to make sure that it was impossible. You will not find that these overlap in any way. So okay, when they don't overlap, they basically become their own units called contigs. This is a word that a lot of you might be familiar with. And what we do to improve our assemblies is that we actually have a third library consisting of long reads. They're basically, you can think of it basically that they're cut out of the genome, and we know how long they are. Our inserts are about 3,000 bases, and we sequence the tips of them, about 150. And then basically we search all the completed contigs and look for matches. And we find a match to create something called a scaffold, where now we both know the relative position of various contigs next to each other, but also the size of the gap. Again, we don't know what the sequence is, that's what the ends represent, but at least we know how much space and how large our gap is, so we can go back later when we have more data and fill it in. And this is, again, repeated iteratively, so here it's represented this way. So you have a scaffold, and here you have another contig, and you have another long pair read that might map this one with this one, or another one, and then basically you continue as compressing it all down, so you have these nice assemblies, hopefully. So we have our assemblies. I managed to produce several assemblies of varying quality. And so the next step then was to annotate it. Okay, we have all the letters more or less, kind of know the order, whatnot, but what are they? It's A, C, G, and T, we don't really know. So that's the next step called annotation using this program. Name doesn't really matter. Basically what this does is that they have a database, the OrthoDB, that has already existing various genomic databases of organisms. So you can choose one, Nematoda, Arthropoda. Well, we wanted Tetrapoda, which is the closest to a turtle that they offer here. We don't want mammals because that's a little too specific. It's again organized by plate. So we choose Tetrapoda, and boom, what do we get? Well, we identified about 300 different genes in our current assemblies, and it can only be improved further. These are some of the genes. You don't really know what they are. It's just, hey, here are some what they did. The annotation gives you a prediction based on current existing ones, what kind of genes there might be. And again, there's still some information missing. We know that EOG09, whatever, it's a zinc finger protein, but we don't know exactly where it came from, like where this information come from. So a lot of you guys have mentioned GenBank earlier. For those who don't know, GenBank is basically a very large database where research groups and studies basically deposit all their sequencing data. So what we would do, we could take these sequences that were annotated and found within our assembly, and then we can basically tell their GenBanks to have their own algorithm, basically use an alignment to look for matches in deposited sequences and see what do we get. And I'm very pleased to say that our closest match was this little guy. It is italicized, good. Chrysemys pictabelli. And that actually turned to be really good news because I went and found a phylogenetic tree. I did not make this tree, I did not have time to do that. But basically, the Chrysemys pictabelli and TS elegans, the red-eared slider that I'm working with, are pretty close to each other on the tree. They're both in the family Amididae, they're both turtles. And basically, it's a nice confirmation that we are doing this right. Okay, that this is the Western painted turtle, Cressemys pictobelli, is the only other turtle that has its genome sequenced. And so basically, where we are right now is we want to improve the quality of our red ear slider to be at the same publishable quality the Western painted turtle is. And then once we have that, a more complete annotation of various genes involved, we can do comparative genomics and maybe hopefully identify genes that TS elegans has picked up from introgression of T. gagei, and that these genes, whether or not they actually give selective advantage to these turtles and their invasion. So that's it. That's all I did. Thank you very much. Thanks to Brian. He's not here today. He was in Cabo, BMXing, snorkeling, skiing. I was making a presentation. Thanks again, once again, heartfelt thanks to Rich, Catherine, Kelly, the whole crew, and all my cohort. Made this not just bearable, but really, I can honestly say, the best summer experience of my life. And I kind of want to do this for a living. Can I come back? And I think biggest thanks go to Joe Russack, and Jim Henderson. This was my first research experience in computational biology. So thank you. You guys were very patient and very helpful in kind of making me into a somewhat comfortable and proficient computer user. So thank you guys. It really means a lot to me. And that's it. Any questions? Really? All that made sense? Oh,
so what's next?
What's next? So I mentioned, oh, I have to do this now, can I? This thing. Okay, I mentioned that we have gaps. So I didn't report on the actual stats, but basically, to give an example, the Western painted turtle, the published turtle genome, has about 2.9% of their contigs unscaffolded, meaning they don't have a partner. We're actually at 25%, which is huge. But that's relatively, I mean, all these programs, they work, they've been done before. There's dozens of organisms' genomes that have been sequenced, so they work. We just need more data, basically, to fill in those gaps so we can make larger contigs and then get more contiguous scaffolds. I think, yeah, I got set up a VPN, so I can actually, if we have more data, I can continue even when I'm back at my home institution.
Yeah, I know just enough about this to be really dangerous, but I was just wondering how much, what is the likelihood that these gaps are going to interfere with your ability to identify a functioning phenotypically expressed gene?
So when we run it through Busco, which is the annotation program, and then we blast it, they actually, when we blast it, it actually tells us how well you match. Okay. So we get a sense, and basically, I don't know what the threshold is, but basically, if there's too much of a gap, yeah, I just won't identify it. But I didn't show it here, but I can always show it later. But you can see there's sometimes gaps when I blast annotations to see against the known databases. So basically, I think the best answer is it's ignored when it uses a certain threshold. That's why we like more data to fill in that.
It depends.
Yeah, it depends. That was the long version. Yeah. Yes.
You mentioned that you did 3 assemblies.
So 4, but yeah,
and
doesn't matter.
What were the major differences between them, and were they all uniformly one better than the other?
So the differences between them, basically the main thing that I did to change them was choosing that Kmer size. And the differences between them, basically, I noticed that when we chose smaller K sizes, we had a larger number of our contigs were bigger, but the total largest contig, the larger the contig is, the more information you get, was smaller, and vice versa. That was basically the difference between the 4. Yes?
And following on from that, what makes one better than the other?
The different programs or the different?
You made 4 different assemblies.
Oh, okay. So basically, 2 of them were using, really quick. What happened to the? Oh, there it is. Oh, geez. Never mind. I don't want to go back, run backwards. So there were 2 Abyss and SOAP, 2 programs. They're basically more or less the same. Jim convinced me that SOAP was a little better because you could give it more inputs and tell it to do more things. But they basically use the same algorithm to build it. The main difference was, again, choosing the K size, the Kamer size.
And it's frothier.
SOAP?
Yeah.
Yeah, I guess you have a lot more. You get a lot more out of it than I
It's
did out
it's
of
not
it.
monotonic. You can't say one is exclusively better than the other. And two, so out of the four, he's got two that are not worth anything, and two that both exhibit some really interesting dynamics.
Because I basically pulled the best Abyss assembly and the best SOAP assembly, basically. Yeah, I used both of them.
And the next thing we do, there are programs that actually take assemblies
Yes. The
and
meta assembly.
make better assemblies for multiple other assemblies. So which it requires what's called the jumping library that was described early on. If you've got one of those, then it uses those to score the assemblies, and it's, it's called meta assembler, obviously.
But the better quality assemblies just had smaller gaps, or?
They have smaller gaps, less gaps, larger contigs,
Yeah.
things like that. Yeah. All right. Anything else? Well, thank you for your attention.
Thank you, David.